I will be brief. Just want to mention some of the main objectives we expect to achieve in this workshop. There are 4 points that we believe it's our priority. I would like to emphasize that this workshop is focused more on people who are dealing with the equipments, who are organizing the facilities, and also, of course, the users. But our main idea here is to discuss how do you organize these facilities, how do you operate multi-user equipment. So that's the first point. So there are a number of issues here. First one, how do you have the adequate, the right publicity? How do you explain to our users how the regulations, how the facility operates, and the accessibility to the multi-user equipment, so that this has to be very transparent, very clear, and so on. A second point within this organization ways is how do we cover, and if you do, and we do, but how do we cover the operational costs of the facility, of the equipment, so that you can keep it operating in a successful way and updating it whenever it's necessary. So this is the first point. The second aspect we would like to discuss, which is very important for me, particularly for my previous experience. What is the strategy of the laboratory, of the facility actually, and the research laboratory which operates multi-user equipment, to differentiate what we call a service-like and what we call a scientific collaboration. Because some facilities don't have this problem because they are built like that, but others, when you introduce a multi-user equipment in a laboratory, a research laboratory, one thing is operating as a multi-user equipment. The other thing is the scientific collaboration you have, and how of course you cannot make this. There are gray zones here, but to understand how to operate this in a very clear way, this is another aspect we would like to discuss. Third aspect we want to see is. That's my previous experience at the synchrotron. We start to see this more and more, and I believe the next machine will be like that even more. Equipments are becoming very sophisticated, and you have to operate them in a sometimes with a continuous feedback from the user, and not always the service becomes more complex, and the participation of the user of the facility with the technical people from the facility has to be in a very, let's say, interchangeable way. So this is something that we have to discuss to understand, because more and more these equipments require very well-trained technical people. And finally, the last point, and equally maybe even more important, what is the right place, the adequate place to install a multi-user equipment? That's something we have to consider very careful. Is the research laboratory, what is a facility, what is a research laboratory, what a research laboratory can do as a multi-user eventually, and what we should actually focus on facilities and develop more and more the concept of facilities in our laboratories, in our universities and research centers in the state of São Paulo. So that's the idea we have for the workshop, and I hope we have enlightening from, I'm sure we'll have enlightening about these subjects from our speakers, and we'll have opportunity to discuss that. Thank you.
So our next step here is that I will give you a brief overview on multi-user equipment in terms of FAPESP, or how we have been working with that in the last 10 or 15 years. And just to bring up some of the challenges related to selecting, to funding, and to following the usage and the implementation and the offering of services in multi-user equipment. So the presentation will go through multi-user equipment, support for infrastructure funded by FAPESP, of which multi-user equipment is a part. Today it's about 30 to 50%, depending on the year. And then a brief demonstration of how other agencies in the world deal with multi-user equipment, from which we can learn some ideas. Some slides are in Portuguese, some are in English, but I'm sorry for that. But anyway, FAPESP supports research infrastructure through a number of programs, which presently are the first one listed in red in this slide. We have the Reserva Técnica, the overhead, which is paid to each institution, which is for the institution to use in its research infrastructure. Of course, the research infrastructure includes multi-user equipment whenever the institution finds that that is sufficiently relevant. That's actually something that we pay attention to, because a measure of the interest of a community in something is how much that community is willing to use the money that is freely put in their hands to be applied in that something. So if you have a lot of money in Reserva Técnica and you never use anything of it in multi-user equipment, then in our view, that community doesn't really need too many multi-user equipment. It's something that is put in the hands of the community to decide how they want to work on that. One difficulty that we, just to mention one that we have been noticing in the Reserva Técnica, is that in many institutions, and that is something that FAPESP does not recommend and does not support, but we have found some cases in which we actually slash the budget, in which the institution, instead of making an internal discussion about which are the infrastructural necessities of the community and focusing in problems, one this year, one next year, and so on, what they do is they try to redistribute the money to each one of the researchers who had grants. That is not what we expect from Reserva Técnica. We expect focusing of the investment, not spreading of the investment. Then, other than Reserva Técnica is now a large fraction of our yearly expenditure. It's probably the fourth largest expenditure every year that FAPESP makes. So the first one is doctorate fellowship, the second one is postdoctoral fellowships, the third one is regular grants, and the fourth is either thematic or Reserva Técnica. In some years it's thematic, but next year it's going to be Reserva Técnica, because there was the Reserva Técnica from CEPIDS, which increased a lot in some institutions, the Reserva Técnica. So it's not small money, it's a reasonable amount of money. Then there is and because it's a reasonable amount of money, we are getting more and more picky about the efficiency of that, the use of that money in terms of research infrastructure, not in terms of happiness of everybody. Those might be different things in many situations. Then there is the multi-user equipment, which is a program for which we have had calls, 2 calls, in the last 10 years, and for which sometimes we identify an equipment requested in a thematic grant that cannot be approved in a thematic grant, but might be approved as a multi-user equipment. Then we separate that as a separate request for a multi-user equipment, which has to follow all the rules of multi-user equipment. Then there is the network, the internet network that FAPESP funds in every institution. Then there is the books, and then there are other smaller programs, but those are the main ones. As I was saying, the last bullet there states that since 2007 we have been expecting that the Reserva Técnica Institucional was created to this end, for the institution to take care of their research infrastructure, so that we will not have those big calls FAPESP had back in the 90s, like infrastructure for everything. Now it's paid every year for the exactly to avoid the necessity of, after 10 years, needing to spend $400 million in infrastructure. So you pay $40 million every year so that people keep their infrastructure rolling. Just to give you an idea, if you add from 1995, we applied more than 1.4 billion reais in infrastructure to this day. There was a steep climb there in the 2000s when we had the infrastructure programs, and then there was the multi-user equipment call in 2010, but overall it adds to 1.4 billion. And for money which is either directly or can be associated to multi-user equipment, we are talking about around 550 million reais in the last 14 years, since the year 2000. The blue bar shows the multi-user equipment expenditures of, it's not expenditures, it's contracts of FAPESP, and the red bar shows the part related to the Reserva Técnica Institucional. So as I was showing to you, the Reserva Técnica Institucional is a sizable part of it. And one of the things that we expect of this workshop and other discussions we have been doing with the universities is that they should pay attention to the Reserva Técnica Institucional. I think I mentioned that already. I've been speaking for 7 minutes. I mentioned that 7 times today. So it's 1 time a minute. That's to make sure that you pay attention to the thing. In terms of multi-user equipment, since 2000, we applied 144 million at the University of São Paulo, 49 million at the University of Campinas, 25 million at the University of the State of São Paulo, 14 million at the Federal University of São Paulo, 12 million Ministry of Science and Technology, which is the sum of their institutes, INPE, CNEPEM, and others, and other research institutes got the balance of the money. In terms of the Reserva Técnica Institucional, the University of São Paulo gets 114 million in the last 6 years. University of Campinas, 34 million. UNESP 27, and then Federal University 16, and the institutes, the state research institutes, 11 million. Well, considering only the multi-user equipment expenditure since 2005, FAPESP spent more than 250 million in multi-user equipment. There is a site, a page in the FAPESP's website, which is at fapesp.br/EMU, where you can see all the equipment and the facilities that were funded. And actually, one of the things that we are now starting to work with the universities is that we need and we want and we will have each university to have in their website a list of the facilities and major equipment, because I'm sure it doesn't happen at your universities, but some institutions and some researchers, and I'm sure those are not you, try to hide the equipment so that other people won't use it, or it will be difficult for people to use it. We actually received already, in some cases, emails from, because we keep track of those websites, and sometimes we send an email to a researcher saying, well, your website doesn't have the instructions on how a person can use the equipment. And then the person answers to us, well, you know, I put that in the 5th page because I didn't want too many people to send requests. So but I'm sure it's not common. Anyway, in the 2009 call for proposals, We approved 170 grants for 167 million reais. And an important novelty in this call for proposals, in this call from 2009, was that we were much more insistent and picky about institutional support than ever before. So for each equipment, for each concession, before the concession, there was an interview here at FAPESP with the PI, with the director of the institution, and with the dean for research or a representative to discuss how is that the necessities to the demands and the things required to keep the equipment running were going to be provided by the university. And each university signed a written statement about that, about the compromises, which is a statement that we use to follow on the usage and the workings of the equipment. Sometimes it happens that a user writes to us saying, I tried to use this equipment, which is in FAPESP's list, and they told me it's not working. So then we go to the PI and say, why is that it's not working? And he says, well, I don't have a technician. And then we go to into the contract, and it says the university is supposed to provide a technician. Then not very good things are going to happen to that institution and to that PI. I can tell you that every institution in the state of São Paulo is able to find a way around those problems. Everyone. We have seen cases in which we made the phone call in the morning to the institution and to the PI saying, why is that you don't have a technician? After 2 hours, we were in the phone with the rector of the university, and by 3 o'clock in the afternoon, there was a technician being hired for that lab. So they can find the ways to fix that. There's no obstacles that prevent universities from solving those difficulties. So institutional support is very, very important. The thing is that not only multi-user equipment, but in many other topics, in almost everything that FAPESP does, our conclusion is that if we want to have internationally competitive science in the state of São Paulo, we need to have internationally competitive institutional support to the researchers in the state of São Paulo. There is no other way around that. There's no simplifying the law, simplifying procedures. None of those things will fix the real problem, which is if you want to have the kind of science they have at North Carolina, in Stanford, in Cornell, in Berkeley, you need to have the type of institutional support they have in North Carolina, in Stanford, in Berkeley, and so on. It's as long as the researchers here spend their time fixing machines, making phone calls, doing accounts and everything, we are not going to have internationally competitive science. So that's how we are working at FAPESP. So unfortunately, it's a problem that FAPESP cannot solve alone, because if we are talking about institutional support, the institutionals have to do their part. So we have been working very intensely with all the universities and research institutions in São Paulo in relation to that. For the equipments that we bought in 2009, most of them are operational and working, but some of them are not, and that relates to difficulties that we have found in institutional support, in construction, especially in the cases in which the institution was supposed to build a place for the equipment, right? You know that it's not very easy to build because of several legal complications, but still it has been 5 years. So we are working with those organizations to see how this will be fixed. Another way in which some, very few, a small number of multi-user equipment has been bought, especially since 2010, is that you know that in the review form for a thematic grant or for a young investigator grant, there is a place in which the reviewer can make a note answering to the question. Is there any equipment in the list requested which qualifies as multi-user equipment? So the reviewer calls our attention to some equipment. The panels here at FAPESP also pay attention to that. And when we find equipment that might qualify as multi-user equipment, what we do is, if it's considered really necessary, if it's not doubling or repeating equipment which exists at that institution, because you know that also when a request is submitted, it's necessary to present a list of the major equipment that exists at that institution. So if you get a request in which you look at the list and there are 9 HPLC equipment operating at that institution and they have 100 professors, and then the researcher is asking for a 10th HPLC, we will first ask the institution, why is that they cannot use one of the 9 which exist there, right? And then if the answer is reasonable or not, we make our decision. That's also an important thing to bear in mind. Many times there is a place in the form for submitting a proposal to FAPESP where FAPESP requires actually a justification for the equipment. And most of the time, the justification that we receive is very close to useless. It is a justification that says, well, I need to buy a stair because the use of the stair will be to go up when I'm down and go down when I'm up. Right? That's not a justification for a stair, right? It's, or I need to buy a PCR machine because I need to use PCR in my research. So justification must relate to, there are so-and-so equipments at my institution, their specs do not reach the specs that I need because in my research the specs are this or that and so on. So we are more and more being careful about reading those justifications. It's in the responsibility of the researcher to justify. If the justification is not reasonable, we will not approve any equipment. It's, the researcher has to write a compelling case to us about the need and the necessity of that particular equipment and about why is that they cannot use equipment which already exists in their institution. That's very, very important. Why is that they cannot use it or they will not use? In many cases, it's very legitimate. There might be a huge line. I have to wait for 4 months. Okay, let's see the list of usage, how many hours the equipment is up every week, and so on. If it's okay, it's justifiable. But we need a real justification, not a number of letters in a paper that don't mean a real justification. So in those cases, when there is this kind of justification, we might separate that equipment and make a special multi-user equipment request attached to that process, which has to fulfill all the multi-user equipment rules of FAPESP. In some cases, now there are becoming many cases, the location of the equipment might be decided by FAPESP. If somebody wants to buy an equipment that we feel will fit best in a facility, we might fund the equipment, but we will send the equipment to a facility where it will be more used, more announced, more visible, and so on, or more efficiently used by the community. Well, I should mention to you that FAPESP has a number of explicit requirements for the institutions in relation to multi-user equipment. From the host institution, FAPESP requires adequate physical infrastructure, technical personnel provided by the host institution necessary for the perfect operation of the equipment. Perfect operation means perfect operation for that research and for other users. It's not that you can say, well, I have 2 students who will take care of the equipment. That doesn't work. Has to be a staff that will take care of that, or a system for training users, which most of the time happen in a facility. Then the institution also has to take care of insurance and maintenance of the equipment through contracts or through an internal system they might have. Some universities in São Paulo have very good internal systems for the maintenance of equipment, So that's very acceptable, to keep the equipment in perfect conditions for at least 7 years. From the PI and the host institution together, FAPESP requires the establishment of a clear and visible set of regulations and procedures for the access. Clear, it must be written. Visible, it must be in a website accessible to anyone in São Paulo or in Brazil who might want to, or in the world, who might want to use that equipment. And use that equipment, the fact that FAPESP funds the equipment doesn't imply that the usage of the equipment has to be free. It's completely reasonable that the usage will be costed. It costs money to use an equipment, right? You have to buy consumables, you have to keep it running, you have to make sure you fix it by the end of the year to keep it running. So those are all acceptable costs that any researcher that asks funding from FAPESP can request in their funding. So many researchers say, well, I need to do, I don't know how many million sequencing, and I will do that in Korea. We don't want them to do that in Korea. We want them to do that at Lactad, at Cefap, at Exalc, at Premium, at one of the facilities here. It's not a difficulty to fund the cost of doing the research. That's something normal in the FAPESP system of funding. But if the facility doesn't charge, then the person will not request the money, and then the facility will not be working after 7 months because they won't have something that they needed to have. Also, the PI and the host institution must organize, either for the major equipment or for a facility, a user committee. A user committee is supposed to oversee the workings, and we progressively are using that. We need, in the yearly reporting of the PI about the funding that FAPESP paid, we need a manifestation of the user committee. The user committee, it's not enough for us, for the PI to say the equipment is working and everything is okay. We need the user committee to say it is okay, to say that users have been provided with decent service here. Users can use the equipment in a line, in a sequence which is reasonable, and we are happy with the way it is, or we are not happy with the way it is. Whatever. They can write whatever they want to write in their review. But there must be a user committee to oversee the quality and accessibility of the services. And we need this web page with all the necessary information for people to be able to plan their research, counting on that facility or multi-user equipment. If you go to this, as I mentioned, to this web page, we have the items that, I think there are 250 items there, around that. There are supposed to be 250, but we found a number which the link is dead, the information is not there, and we are constantly working with the PIs to have that fixed, as I mentioned to you before. And there you can search by type of equipment, for example. You can go there and you will find that there are 13 mass spectrometers of some kind scattered around the state which were bought in 2010. And then you can go to the web page of each one of them and find what has to be done to use that, to be able to use that equipment. 12 mass spectrometers, 9 cytometers, 9 spectrometers, 7 sequencers, high-end sequencers, confocal microscopes, and other kinds of equipment, NMR, everything is there in the website. FAPESP is encouraging the organization of multi-user equipment in facilities, and that was an important point in our 2009 call for proposals, in which we explicitly wrote that proposals related to facilities would be especially valued in the call for proposals. Several facilities were created. We have a number which says more than 50, the ones that we listed as self-declared facilities. We are now in a process of analyzing each one of them to see if if their definition fits with our definition of a facility, right? So it could be less than 50. We have seen that in many of those, and we have visited some of those, there is a progressively professional management. They hired excellent staff to take care of the equipment, to help the users. You will see in some of the presentations today how does some of those work. They are having increasing visibility to the point that some of those facilities are already at the point in which we are getting complaints from users that say that the delay for use is too long. We are trying to understand if that is so or not. But many times a person sends a project in which they want funding to sequence whatever in France. And then we send a letter to the PI saying, well, you should try those 3 places before. And then they write to us and say, well, I went to the 3 places, they say I have to wait 4 months. And then we go to the 3 places and see what's going on. Right? Sometimes it is like that, sometimes it's not, sometimes there is a misunderstanding and so on. But anyway, the facilities are really improving. This is something that, if you think about 10 years ago in São Paulo, nobody talked about facilities. The chemists did. In the chemistry institutes, they had what they call Central Analítica. At the Chemistry Institute here in São Paulo, in Campinas, I remember Fernando Gallenbeck was one of the persons who organized that there, and in some other institutions. Somehow the chemistry community worked on that much before here in São Paulo, much before other communities. But 10 years ago, you didn't talk about a facility for sequencing, for omics, for spectrometry, for this and for that. And now you have for a good number of those. And they are really working and moving ahead to be facilities that I believe at some point they will be able to offer services outside Brazil. So that researchers from other countries, for example, in Latin America, will buy services in the facilities that we have here in São Paulo. They are getting increasing institutional support. The leadership in the universities is noticing that there are effects, there are scaling effects in supporting a facility as opposed to having to put a technician in each one of the labs that has a major equipment, because then you can divide much better their time. And as I mentioned to you before, concessions made by FAPESP of multi-user equipment might require that the equipment be located at a facility, unless there is a very special scientific justification to convince us that the equipment has to be somewhere else. Just to give you an idea of the difficulties there, of the 250 links that we tried in January, each one of them, for 75 the web page was not found. For 101, there was no information about how to use the equipment. Those are the ones that you have to go to the 10th page to find, well, send an email to whatever. For 97, there was no indication of the procedures. The 101 is not how to use, it's information about the usage. It has been used 100 times last month or 10 times last week or so on, because that's something that we require to make to give an idea of the possible delays in an objective way, not in a way in which is a sentence written by somebody saying, well, there's too much delay. If there's too much delay, we want to know why. Why? You need to understand the numbers. The machine has been working 8 hours per day, but it could work 12 or it could work 24, right? So it's something that scientists can understand. It's not for politicians, it is for scientists. And 64 presented the required information. So we are working with the balance with the remaining ones to have them do it correctly. And it's something that we expected, because of course in Brazil there's not a tradition of facilities, of sharing equipment, of sharing major equipment. The tradition has been, I would say, until a few years ago, maybe until 2000 or 2000 and so, except for very large things like a synchrotron light source. Okay? But for other things, the tradition has been to each researcher wants to have the thing locked in his lab. Even if they use the thing like 3 hours per week, but they want to make sure they have. And it's not that it's something people do because they are bad. It's something people do because they want to survive. Because many times if you put the equipment in the common place of the university would not take care of that. And then, okay, you buy the thing, you put it there, and next month you go there to use and it's broken, there's something missing, and so on. So I'm not saying that there were not good reasons for this kind of bad behavior. But we want to change that. We want to fix the reasons because it doesn't make sense. You cannot have a mass spectrometer in each one of the laboratories. No country in the world has mass spectrometers in a researcher's laboratory. They have a mass spectrometer in a central facility and other equipment too. So that's the thing that we want to do to make funding of research in São Paulo more efficient and to make the usage of the results of that funding, the equipment, more efficient for the whole community. So that's the big objective of this workshop. So with this, I will just spend a few minutes, 5 minutes here to show you how some other organizations which do the same type of work that FAPESP does, deal with that. For example, the National Science Foundation. Everybody has heard about the National Science Foundation. They have a program which they call the Multi-User Research Instrumentation, MRI, which has many things which are very similar to the way we deal with that. For example, the researcher has to demonstrate that there are 3 or 5 or 7, I don't remember the name, other researchers who have grants who need to use that equipment, the same thing as you do when you submit a multi-user equipment when we have the calls. Now they have some things which are different. 2 of those are highlighted in this slide. First, they have a submission limit. Each organization can only request 3 proposals every time they have a call. And that is associated to the fact that they only work with multi-user research instrumentation on a cost-sharing basis. The university has to pay part of the cost of the equipment. So universities will not be able to pay for 100 equipment. So 3 is a reasonable number for them. In this case, the National Science Foundation asks the institution to cover 30% of the total project costs related to the multi-user equipment. And they give awards which are from 100,000 to 4 million for they have acquisition and they have also the development of instrumentation. This is something that we do not have here as an organized program. We receive proposals which sometimes we fund for the development of spectrometers, of detectors, of sensors and stuff. But it's not part of the multi-user as they consider that. But you see the range is more or less the range that we use, and the main difference is this relating to the cost sharing. Another organization, and this is representative of what happens in the other research councils in the UK, is BBSRC, which is the Biology and Biotechnology Research Council of the Science Research Council of the UK. It's an organization with which we have been interacting a lot in the last years because now we have a good number of researchers in São Paulo who have proposals together with researchers in the UK. And one of the things that came to our attention was that the proposals in the UK, they never have large equipment. They never request large equipment. If they request an equipment, maybe 10, 12,000 pounds, not more than that. They never require a big thing in the projects that we have seen. And we learned that, we understood that by looking at their treatment of equipment. What they do is that equipment below 10,000 pounds, researchers can request in a normal funding grant. Even though most of the time they don't, but they can. They tend to favor fellowships and travel and consumables in their grants. So for equipment below 10,000 pounds, they can request in a grant. For equipment between 10,000 pounds and 136 approximately 1,000 pounds, and this number 135,688 is a number that comes from the European equivalent to our 8,666 law. They also have a law for buying stuff, right? It might surprise you, but Brazil is not the only country in the world in which there are laws on how you use public money. So in the European community, they have a legislation that says if the equipment is below this value, you buy in a simpler way. Doesn't mean it's simple, it's simpler. If it's above this value, you buy in a very complicated way. There's several pages of regulations that have to be understood. But anyway, what I highlighted there is that BBSRC will contribute no more than 50% of the final purchase price. So they say, if you want an equipment, first convince your institution that they are willing to pay part of the cost for it, that they really need this thing as much as they are willing to pay for it. If the equipment costs more than 136,000 pounds, then they will fund only where there is a clear case that the equipment will benefit a range of research to encourage a culture of equipment sharing. So they too are worried about encouraging a culture of equipment sharing. And single capital items of equipment over 136,000 will normally be funded at 50% of total cost again. Cost sharing of the equipment. Cost sharing is something very common in many cases. Other organizations do not use, I should say. For example, NIH has a program which offers every year maybe $40 million, which is very competitive and very complicated to make a proposal, in which you can request large equipment without cost sharing. But it's $46 million per year out of the $30 billion of NIH, right? It's a very, very small fraction of their funding. So you see that buying large equipment is not something that research groups do every year or every 3 years or every 4 years, even in rich places like the UK or the US. This is some of the misunderstandings that seems many times that our scientific community adopted, that you only do research if you buy a major thing every 4 years. It's not like that. You buy stuff to keep your things running, and you have facilities that you can use, and the institutions take care of having the facilities running and working efficiently so that people who need to use them will use. Well, we would like to be in a world like that. We are not. But we are trying to learn. And this workshop aims exactly at the idea of learning and discussing among us and listening to people who have done that. That's why we invited Piotr to be here with us today. Thanks for coming, Piotr, from North Carolina. He came down just for those 3 days to be with us, to make a presentation, to have a discussion with us later today. He runs a very interesting facility for sequencing in North Carolina, University of North Carolina in Chapel Hill. And so we want to learn to make our system more efficient in terms of better usage of our money, of our funding for research, so that we have access to equipment in an efficient way and in a way in which researchers can have access to equipment which is modern enough to make them competitive. So that's the whole idea of this discussion, and I thank you very much for being here today. Thank you.
First, I would like to thank Professor Brum for the invitation to come here, and I should also apologize for the slides that is all in Portuguese, but I'll try to speak in English. So this first slide is just to show the what Brito had said, that to improve the efficiency of the investment of FAPESP in those multi-users equipment is absolutely essential to have the support of the institution. And this is the laboratory, the building that was built by the University of Campinas, just to house the facility that is called LACTAD, that means that Central Lab for High-Profile Technology in Life Science. And this was a decision of the university to set up the facility because when we ran in 2000 the genome program here at FAPESP, and we had a kind of multi-user lab in a kind of network, and a kind of organization that took care of helping scientists in analyzing the data that was generated for the sequencing that was produced. And this, the way the system was organized is a kind of super facility, so that, for example, bioinformatics was organized in a way that every student and full professor could have access of everything, all the data at the same time. And then the idea here is of this facility, University of Campinas, is about the same. So in 2009, the University of Campinas decided to set up this facility with infrastructure, personal equipment, and lab facility for to make available to researchers at the state of São Paulo the access of technology for those high equipment, high-profile equipment, and in the life science. So this facility that is called Central Lab for High-Profile Technology, it was the concept is that it should be the administration should be a professional in a professional way. So with a team that do the administration and run the facility in a professional way. And the team has no, let's say, in the sense that people, for example, are more familiar with a system where people need to publish papers and be involved in a kind of academic research. So they do research, but they do research to improve the techniques and methodology that is run in the facility. So the compromise of the facility of the team is to deliver results of high quality in efficient way in a time, reasonable time that is contracted with the users. So an additional focus of LACTAD is to train people systematically, mostly in bioinformatics, because this is a lack of expertise, not only in the state of São Paulo, but in all the country. So LACTAD is putting a lot of effort in training people. So our mission is to make available to the community the technologies and services in the area of life science, and make available for the users of techniques available, and also to help students and scientists to design to help them to plan the experiments that they are doing, because this is one of the major problems with the facilities, because sometimes the researchers comes or the students comes with not very well planned experiments, and then they want the data, and if the data doesn't come with what they expect, they think that the problem is with the machine, but it's not. It's most of the time it's planning. So we help planning, we help sample preparation and processing, and we also help we offer for students to come sometimes to learn how to prepare samples or extract DNA, extract RNA, and things like that. So how we operate. So the University of Campinas decided to build a 2,500 square meter lab just for this facility, and also to provide the technicians and administration to run the facility. And this is what we are today. So we have 17 places that was offered by university. We have a kind of board members, and this is not board members in the sense that we are going to decide or change direction in terms of what kind of science should be done at this facility. This is just administration. So we have board members. We have we hired general administrative. It's not administrative. It's kind of general manager that is Sandra Koroshenko, that is here, and we have a secretary and IT support analyst, and then we provide we work in 4 areas: in genomics, bioinformatics, proteomics, and cell biology. And for each of these 4 different areas, we have a team leader. All of them is already hired by the university, and we have the specialists. We are now putting effort to contract the last 5 or 6 members that still the university provide us with the possibility to hire those people. So this is why we are going to be, perhaps by the end of this year, with 70 people to run all the equipment and the technologies in the facility. Those are the board members. This lab is not linked with any department or institute. It's linked directly with the Dean for Research. So the Dean of Research, this is to keep, the compromise of the university with this facility, because the university decided that it's so important to have this facility running in a very efficient way, professional way, that it's linked with the Dean of Research. And this is the board members: Professor Sara from Medical School, Carlos Henrique from Chemistry Institute, Gonzalo from Institute of Biology, Zanoni from Computer Science, and myself. I'm here representing this member. I'm not the coordinator. The coordinator is Sandra Koroshenko, that is the professional that was hired to run the facility. I'm only representing the board members. And this is the team. Sandra Koroshenko is the general manager. Wagner, Leandro, and Osvaldo are the bioinformatics. Felipe runs the cell biology machines. Vanessa is the leader of the proteomics, and Susie is the leader of genomics, and Luciano is the secretary, and Felipe have a fellowship in TI. So the university not only gave the resource to hire people, but also provide fellowship. The Dean of Research provide fellowships for bioinformaticians, students for bioinformatics and cell biology. And Felipe is one of those students that is working together with Roberto in order to help us with all the IT issues in the center. So, where the money comes from? So, university built this lab facility, the building, 2,500 square meter. The university also is buying all the infrastructure, the lab bench and the offices, furniture, and the university also is give the money to hire the general manager and 16 technicians and specialists. So we got the money for sequencing and the large equipment, as Brito showed, from multi-users. We have sequencers, first generation, second generation. We have machines for bioinformatics, cell biology. And the Dean for Research also provide fellowships for bioinformatics and cell biology. We also have approved from FINEP CT infra equipment that we are buying mass spec, and we are investing in automation because all this to gain in efficiency, we need to have automation in lab preparation. But most important is that we have our focus is to be self-sufficient in perhaps not so, because when we set this facility, we were thinking that we could be self-sufficient in three years, but it's not possible in our condition. But I think that we can sometimes be self-sufficient, so we have a system to generate cash in terms of charging for all the service that we provide for the scientific community. How do we do this? Well, this is I'm not going to detail this, but Sandra and the team, they organize a system that we can estimate the cost of buying equipment and depreciation of equipment and all these things, and so that we can reach, in a formula that we can estimate the cost, the real cost of any single technique or procedure that is done at this laboratory, so that we can estimate the cost, and then we can charge the real price for the service that is provided for the community. As I told, we have genomics, we have Sanger equipment and high-seq equipment. We have two high-seq Illumina. We have cell biology, confocal, immunoassay, flow cytometer, proteomic. We are running proteomics together in collaboration with Chemistry Institute because we don't have yet the mass spec in the laboratory. So we are in a process of buying one when the CT infra from FINEP, when the money comes. And but we have some equipment for protein purification, and we have bioinformatics. So we have set up equipment, servers, and all the necessary equipment to run to help the students and research not only do the sequence, but also we help them when they need. We help them to analyze. This is very important, and I think that this is the largest, let's say, weak point in life science in using genomics in the state of São Paulo and in the country. So we are thinking that we need to improve or to increase the number of people working in bioinformatics. Sometimes the students are research, they want that we not only we analyze, but we make the tables and figures and graphics. And sometimes, can you also write the paper? This is because, there is a lack of knowledge in bioinformatics. So this is our equipment, and Vinicius and Susie, they run the high-seq equipment, and Janine, that's now at maternity license, and she runs the cell biology together with Felipe. Janine is the leader. Felipe helps with people, and this is very important because people comes with samples, for example, to do confocal microscopy, and they have the support of these two guys here that help them to get the best image possible with the machines that we have at the lab. And this is the confocal microscope that we have. Proteomics, as I told you, Vanessa is running in collaboration with Chemistry Institute, but we are in process of Bio1. This is the equipment that we have for bioinformatics, servers, large, big equipment for bioinformatics. This is our website. All the service can be ordered through our website. We don't, let's say, we don't answer an order coming from, well, we do, but we encourage people to do, to order the service through the website. Most of them send, not most, but few of them send through by email, but we encourage, and in this web page you have all the instructions, all the description of the equipment, the technology that we do, the technology that we don't do, can be accessed through the website. And this is important also because through the website where we can do all these statistics and counting of what people are asking from us. And this is since we start. We moved to this new building quite recently because when we sent the application for multi-user at FAPESP in 2009, we start building the new building. And building in our institution today is something almost impossible. You need to work very, very, very hard to have the building constructed in a reasonable time. So the building was ready at maybe the end of last year. And we are now providing service for many institutions, UNICAMP, ICB-USP, Faculty of Application, Medical School, Food Technology, Institute of Biology, Institute of Chemistry. For the University of São Paulo, many researchers that have big projects financed by FAPESP, University, Unesp, Botucatu, CNPEM, Institute of Agronomic Institute of Campinas, PUC in Campinas, Universidade Federal ABC, UFSCar, but we do also, this facility is open, you know. We don't do preference for the University of Campinas, so we just follow the list of order. We follow the list, and sometimes we have, for example, here from UDESC in Santa Catarina, UENF, Rio de Janeiro, Universidade Federal do Rio de Janeiro, Universidade Federal do Rio Grande do Norte, Viçosa, but we also do research for companies, Braskem, ChemUnion, 3M, that sometimes ask for the service that is done by the facility. So this is our finance status. We have generated about 850,000 reais that we use, to run the facility, mostly to buy chemicals and kits for mostly for next-generation sequencing and all of this. And the idea is to have a cash flow system working so that we can be self-sufficient, at least for generating all the costs and maintenance contracts and all the things. So university provides us with people, FAPESP with the machines, and we make the money to run the facility. This is the idea. We have the training course that is offered every semester, in different and there is a lot of demand. We offer 25 places, and sometimes we have 200 registration. And we run every year also an international meeting that's this year we will be at for August on advanced topics in genomics and cell biology. And we have been mentioned in a different media system. And thanks very much for the attention.
So, good morning, everyone. I also like to thank Professor Bruno and FAPESP for the invitation. It's very nice to be here and share our early experience and learn with the others to improve the quality of our core facility. I will speak on behalf of Professor Menck. He's the leader in our core facility, but unfortunately he won't be able to be here. So I'll talk about FAPESP. We don't have a very nice translation, so I will call just our core facility at the Institute of Biomedical Science at USP. So just to give you a brief timeline of the creation of the FAPESP, back in 2008, our former director, Professor Rui Curi and Benedito Correa, came up with this idea to have a place to gather all this multi-user complex equipment and to find a way to boost the quality and the research of our institute. So later on, we are very lucky that FAPESP launched MU grant proposal. We are financed. We were one of the core facility that was granted. This was later on in 2010. And from that on, we start thinking about a way to be as sustainable as the lactate was showed before. So initially, the Institute agreed to assign 4 research assistants. This was very important because we built up from ground zero in our core facility. Later on, we find out the ways to get being financed outside from FAPESP. So we're very lucky that CAPS with the Pro equipment gave us the cell culture facility. Later on, a new fax machine, and also we have some grant proposal from individuals that got the grant from FAPESP that allocate the equipment to FAPESP. And finally, we start having a feedback from the university. USP has launched 2 different pro-infra grant proposals. We are lucky to get all of them, so we got being financed twice. And also they agreed to give us one administrative staff that is here, is Mauricio. So we are start having a nice structure to run a core facility. And this year we have our regiment that was approved by our assembly, and also we will show later on, I will have all the committees to run our core facility. So where we are here in ICB-4, who you know, the University City, USP, we're very close to the zoo the vet school at gate 3. So this is the ICB-4. We are on the ground floor. We are allocating an area of 120 square meter. It's 1 floor from lactate, but you are very welcome to visit CFAP. We have very different rooms. Each room is allocated to 1 core facility, and I will show you that. So when we thought about FAPESP, we thought about the most useful core facility for our institute and from the University of São Paulo. So we established 4 different core facilities. Each one has a funny name that Menck named while he was thinking about our core facility. So the first one is Fluid, is our image core facility. We have our fax sorting machine. This is run by Fernando Pretel. He's also here. We can do cell sorting and single cell sorting. This was the first, it's a very useful machine for cell biologists, and was the first one that our institute received. So it was the first core facility that was open for the whole community. And it's used from July 2012 until now, over 150 experiments for more than 40 research groups. Fernando Pretel is also responsible to run the in vivo image system. We have two equipments. We have the IVIS and also the FX Pro. So you can do luminescence and fluorescence, as well as do bone densitometry and X-rays. So almost 9 experiments have been done from the start in 2013, and this has been a very good approach. We have now papers being published using these techniques by the users of CFAP. The second facility that we have is the confocal, is our microscopy core facility. So you have a multiphoton confocal microscopy that we can do live cells, even live animals. The laser can go deeper in a tissue, so you can see the movement of the cells and everything. And also we have a high throughput content microscopy, an in-cell analysis. And this facility is run by Mario. This is a high demanding core facility. We are booked every single day since the beginning. We stopped while the equipments were broken, but we run almost 200 experiments with this facility. So we also thought that besides to have multi-user equipment, we should have something that we thought that might be useful to have in a core facility. For instance, we thought that maybe a room dedicated to work with viral vectors, lentivirus, adenovirus could be useful, because this needs to be a biohazard level 2, and if you want to build one in single department, it would be very expensive and also a little bit dangerous. So we built up this facility, so it's also run for a while by Mario. So we have a hood, incubator, everything just ready to work with this virus. So the third core facility is our biomass. It's run by Fernando Almeida, he's here too, and Claudio Angeli. This is the most complex one. We have 4 different machines. We have a multi-off-off, an Orbitrap, a LC MS3 quadrupole, and also a BioCore, and we can run different kind of experiment. Just for single proteomic analysis, for a whole proteomic assay, to quantification of small molecules. Every time someone wants to quantify, we need to establish the methodology. So it's a very complex one, but we are being very lucky with these 2 guys to run and identify, for example, this is a project from the parasitology department that identified the first time the proteins from one mosquito. So if you work hard and focus, as Paulo Arruda has said before, we can achieve nice data. And finally, we also have our next generation sequence facility. It's run by Tiago, Souza, and Laudicea. They start from, as I said, from 0. They were the first research assistant that were hired by us, so they help us to build the facility. We have a Solid XL and a Solid 4, together with a MySec, and also the equipment to build up the library, and also to analyze the quality of DNA, RNA, so we can do whole genome, exome, transcriptome, even SHIP-seq. Just to give an idea, this is one of our data that they could identify a mutation in one disease, in Xeroderma pigmentosus, and since the beginning they all work with almost 60 research group in this core facility. So besides all these equipments and the cell culture system, we also have a conference room. In this conference room, we thought, as Paulo said before, that all the users, they want to discuss with us the way that the experiment will be the best, the most optimized as possible. So we can discuss with them. We invite the companies to present the technology and to explain how to use the machine, and also we have a workshop that we call hands-on, that we have half theory, and then we go to the machine, and then the user can actually see the machine and touch the machine and use the machine and discuss with the technicians from Brazil and even international what is the best way that the equipment could give as data. So we have more than 40 talks so on, just a nice example from the beginning in 2011 to just now in 2014. So another way that we thought that FAPESP-USP could help the scientific community is to allow a physical space and actually a very nice, creative, scientific milieu to allow some companies to bring their machine as a demonstration and they install the machine there, allow people to come over and to see the machine, to use their own samples for free. And then we have quite a lot of companies that came out with different component equipments like flow cytometry, confocal microscopy, machines to isolate and manipulate the RNA and DNA, and also to do the hands-on course, as I said. So we are very aware that something is theoretical, but something is practical. You have to use, to combine both to achieve the best data from the user. So we are trying to do this at FAPESP. So we also have our website. You can log in and you can see all the details from each equipment are described there. Is in Portuguese for the moment, but you can use the English, like Google Translate to English. You can see what's going on. And in this website, you can have the booking system. So the book system should work at the same as Paulo said. So you can log in, I'm going to show you. And in the book system, you have all the details from each equipment, the cost from the equipment, and how you have to pay for them, as he questioned before. So you have to log in, you're going to fill a form. This is very important for us to identify the user, and then we're going to prepare the report. We're going to need who you are attending at FAPESP. Unfortunately, we're still in the process to see if it's working. There are no bugs because we put the payment inside the system. So the user go directly. They know how much money they have, they can use. They have to generate the form so they can pay in the bank. Sometimes they need to put the number of the process in the form. So everything is being setting up so to be as less painful as possible for the user. For now, what we have is a description of the machine and the equipment and the cost and the way they should pay. They should read that, agree, and then they fill a form and send by us. So we book the system, as Paulo said, is the first that arrives, the first that will be run the samples. This is very important because since we have such a huge amount of equipment, we need to know what the samples the guy are bringing to us. They cannot arrive with 100 million cells is just to isolate something that is less than 0.1%. So we need to explain that how they should deal with the samples before arriving so they can get the samples, the best result in a short period of time if you consider live cells. So this is all in this kind of form. Up to the present moment, FAPESP has helped more than 17 different institutions. Inside even each institution, there is several groups from São Paulo, even at outside São Paulo at Federal University of Rio Grande do Sul. And some companies are coming to us, but we need to deal with some bureaucracy inside USP to deal with these companies. As I said, we just have a regiment that was accepted by our assembly. So we have an executive committee that is led by Menck. We have the user committee from people from they are already been using the FAPESP. We have our officer that we can send us suggestions and critics so we can deal with this to improve the quality of CEFAP. So you have two email addresses that you can use to talk with us. Finally, I think since this is an opportunity to present this very early experience, we have to share some anxieties here in our words for the near future. We are not as lucky as Paulo before, so we don't have any bioinformatics at FAPESP. We are there, absolutely looking for them, but this is something that we are worried because we are generating just a huge amount of data, and this data needs to be dealt with and also understand in a multi-user way. So with mathematics, physics, biologists, and everything. So this is something that we are having problem at the moment. Of course, the human resource has been always an issue for us. We are very lucky since the beginning that we have our research assistant. All of them were graduated. They understand what they are doing. They help a lot the user. But we only have 4, at least 5. And we are waiting for 2 more, and this is not coming for the moment due to the situation of our university. And we know that as compared to 16, we need more human resource, but not only one, but someone that can understand how a core facility work. Our machines now are losing their warranty, so we are worried because these are very complex machines. So if you break up, for example, it's a laser is off in the confocal microscopy. We are dealing with users. They want the data, they want the results, so we need that the company give us a feedback very early, and this is made some concern for us. Some of the techniques are become outdated. The next generation sequence, it goes really fast in terms of techniques, so we are worried about that. But we are trying to think how we can be as sustainable as possible in this near future. So I just want to thank initially the former director and vice director of the Institute of Biomedical Science, Professor Rui Cura and Professor Benedito Correa. They have the idea, and of course, after that, we run to get to establish and to get funded. Our director now, Professor Jackson Bittencourt and Professor Luiz Ferreira, and of course our research assistant and administrative staff, então Fernando, Maurício, Fernando again, Cláudia, Tiago, Susan, Mario. And of course this was only possible because we were funded by FAPESP. It was a huge amount of money that FAPESP supported CEFAP, USP also with this Pro Infra grant and CAPS. And we use the foundation of USP to deal with the payments and everything, so we have to thank them too. That's it. Thank you.